Thanks for coming back. Thanks all you guys for coming. You've asked a lot of really great questions so far. And so now I'm going to talk about how we're putting together all these things that we've learned about so far to do a quantitative proteomics experiment, because we're not just so interested in a list of proteins, we're interested in how these proteins change in biology. And I hope that by the end of this, any of you could come to us as collaborators and have a sense of the tools that we could use to do quantitative proteomics experiments, and it will spark your imagination for new proteomics experiments that maybe we haven't thought of, or ways that you can apply proteomics techniques to your particular questions in biology. Just give you this sense of some of the possibilities that are out there. So I'm going to talk a little bit about how quantitation developed in mass spectrometry, and it really came from analytical chromatography as a starting point. And Steve mentioned a little bit about stable isotopes earlier, and these become incredibly important in quantitative proteomics. So we'll touch on that again. And then you've probably seen or heard lots of different acronyms in quantitative proteomics: SILAC, iTRAQ, TMT. There are new ones, pretty much every month if you read the journals. And so I'll try to decode some of that for you and let you know what are the key concepts behind that. And then you may have also heard about something called spectral counting, which I call non-quantitative quantitation, because it's probably the most irreproducible form of quantification you could ever hope to achieve with a mass spectrometer. And then how we use some of these techniques to put together the stories from individual peaks and numbers that pop out of mass spectrometers into meaningful biological information about the proteins that you care deeply about. Okay, so it's important to remember and be reminded of the fact that in our modern era that most mass spectrometers are fed by chromatographs. Chromatography, I'm sure most of you all know, you have a column that has some kind of packing material, stationary phase inside, that has some kind of functionalized chemistry in there. You put stuff onto this column, and the properties of the interactions between that stationary phase and all the molecules in the sample should allow things to separate in time as you pump solvents across this column. And then analytical chromatography will typically have something like a UV or visible wavelength detector at the end of the column, so that as different molecules come off the column, you can get peaks at a given wavelength of the spectrum that you're looking at. So that's the chrome in chromatography, is the color or the wavelength that you're monitoring to detect a given analyte. And there's some common ones. If you look at 260, you're looking for DNA. 280, you're looking for protein or specific amino acids in protein that actually absorb at given wavelengths. Or you could put Comassi blue dye across a chromatography column, and you would see a nice big peak at 595. So you get the sort of trace in time of the chromatography experiment, and things come off. And then what we want to do then is try to figure out how much area under this curve there is. And it's the basic premise of analytical chromatography that the area under the curve of a trace in a detector is proportional to the amount of stuff that is there. So if we can integrate this area under the curve, we can figure out in a given sample how much is there, especially if we compare it to a series of standards that we've injected into the chromatograph of known concentrations of the molecule that we're interested in, or a series of molecules that we're interested in. This is the backbone of analytical biochemistry. So what does that have to do with proteomics at all? So mass spectrometers essentially let you be greedy. Instead of monitoring just one wavelength of light or a couple wavelengths of light, we get to monitor the whole spectrum of mass to charge ratios of stuff in an experiment as they evolve over time. So if we imagine that this picture was broken up into discrete points across the time course of an experiment, and we could draw little dots every time we sampled something, in a proteomics experiment, you get something like this at almost every time point of the experiment. And so this is just a single mass spectrum of now intact peptides from a typical LC experiment. Now we have to figure out how we can quantify all the stuff that's in here as the experiment evolves. So what can we do? One very popular thing to do is to make something that's called an extracted ion chromatogram, and that's sort of like making a trace, just like in a UV-vis chromatography experiment, around one ion in particular. And Steve mentioned earlier how high resolution the instruments that today's proteomics experimentalists use. And so we can draw a very narrow window around one particular mass-to-charge ratio. And let's say that this particular mass-to-charge ratio belongs to the peptide that we really care about. And we can plot the evolution of intensity of that thing in time. So you can't see it yet, but we're going to be following an ion at a mass-to-charge of 655.36. My new favorite peptide, and it's somewhere down in the grass at this moment in time where we've sampled it, and we can't see anything, so we'll put a mark there. And then at some point a little bit later, this ion starts to emerge out of the grass of these other ions until we can draw something a little higher, and then it gets higher yet, et cetera, until now it's starting to go back down. It goes down a little bit more, and now it's back down into the grass. Well, we can do the very same thing that an analytical biochemist would do, is basically draw a line, a smooth line to connect these points, and then integrate the area under the curve. So that's good. This is a very good first step at quantification and something that we rely upon a lot in proteomics experiments. So there are some drawbacks to it, however. The signal, if we look at the signal, again, we're tracing across this ion of 655. It's like very jittery. It's not that nice, beautiful, smooth peak that I made up for the example in the given slide. So the instantaneous variation in that ion flux through the instrument is kind of high. But we can do stuff. We can smooth it out and do stuff like that. However, if we want to inject a series of samples across a long period of time, days, weeks, or if we want to do some today and do some next month, there's a bigger problem that our sort of day-to-day and long-term instrument reproducibility of the amount of signal for any given ion is not very good. If we want to compare a bunch of samples together and we have to digest them all separately with trypsin, there's biochemical and experimental variability that we have to deal with. So that's another problem that we would deal with if we just wanted to integrate all these areas under the curves. And this was also touched upon a little bit briefly in an earlier presentation, but every peptide in the proteome has its own response factor. I'll call it response factor, or ionization efficiency is the technical term, meaning that you could put equimolar amounts of two peptides into an LC-MS experiment, and one of them could have a whopping huge peak of 1 million counts, and another one could have a very small peak of 1,000 counts. Yet these two things are exactly the same amount. This is this ionization efficiency that plagues us in mass spectrometry, and why we don't have the magic solution to say, hey, can you shoot that in there and tell me how much of that thing is in there? Because we don't know. You can empirically determine these efficiencies for peptides on a one-by-one basis. It's very time-consuming and expensive. And even if you were to do that, you would still butt heads with something called matrix effects, and that is when we put all these things into the mass spectrometer at the same time, it can have sort of hard to explain effects of either suppressing certain ion signals or augmenting certain ion signals. So if the matrix and background of your sample is not exactly the same from experiment to experiment, it is very hard to use simple classical analytical biochemistry chromatography techniques to quantify peptides and their proteins in biological experiments. So it would be good if we could have some kind of standard or calibrant that we could introduce into our experiments to kind of make a lot of these issues go away. And this is where stable isotopes come in. So I think of stable isotopes as nature's gift to mass spectrometrists because they enable so many quantitative techniques. And we talked about this before, that if you look very closely at a region of a mass spectrum, you can see peaks that have some kind of uniform spacing, and these uniform spacing corresponds to the number of C13 atoms that might be present in a subpopulation of a given molecule. So what this is basically showing, let's say that we have 1,000 peptide ions inside the mass spectrometer at the same time that belong to this species at 655, our friend at 655. And we kind of know that it has a whole bunch of carbon atoms, and just given the fact that there's this natural abundance of C13 in nature, we can know a priori that some of those molecules in our ensemble of 1,000 are going to pick up at least one carbon-13 just by its natural abundance, or 2 or 3 or 4, and we can use that to determine the charge on this molecule. But then we can also use these stable isotopes in more clever ways to affect quantification. So, for example, what happens, we all know that proteins are made up of amino acids. So what happens if there are amino acids that have stable isotope atoms introduced into them on purpose? So here we have an example of arginine, which can be labeled with stable isotopes in a few different ways. So, and these are just 2 examples. Here's your normal structure of arginine, but if we turned all the carbons in this molecule into a C13, we would get an arginine that were 6 Daltons heavier than this kind of naturally occurring one. Or if we labeled not only the carbons but the nitrogens, then we would get something that was 10 Daltons heavier. So we can now have 3 different versions of arginine, light, medium, and heavy. And if we look at a mixture of peptides, say 1-to-1, that have the light and the heavy version of this arginine inside the mass spectrometer, and here we're dealing with something that has a single charge state, and we can see that now the mass spectrometer allows us to see this 10 MZ difference, and in a charge state of 1, that's a 10 Dalton difference between these 2 things. So if we had a mixture of proteins that contain light and heavy, we could use the mass spectrometer to deconvolve which was which just by the fact that they have differing masses. So one very clever fellow exploited this. This was Xiaoan Ong, who had been a staff scientist here in the proteomics platform. And he said, well, this would be really hard to make all these proteins with stable isotopes, and so we'll just let the cells do the work. We'll take cells and we'll feed them with these heavy amino acids, and we can go from being in this sort of state, and then let's pretend this is a protein. If you make up growth media that has these heavy amino acids in it and take out all the light ones from it, and you do this for a bunch of doublings, maybe 6 or 7 times, what you get out at the end is a protein that is literally a little bit heavier, a little bit more bloated than its kind of light counterpart. But otherwise, it's biochemically equivalent in the way that the cell continues to function, in the way that it will behave biochemically in vitro, in proteomics preparations, and inside of the mass spectrometer, except for the mass. Okay? Now, we like to feed cells with lysine and arginine because trypsin cuts at lysine and arginine. So if we label these amino acids, we can guarantee that almost every peptide, with the exception of maybe the C-terminal peptide of the protein, is going to bear one of these labels. And so this now is what's known as SILAC. It stands for Stable Isotope Labeling of Amino Acids in Culture. And so the basic idea of any SILAC experiment is that you start with, say, 2 dishes of cells, one that you've grown in light media, one that you've grown in heavy media, that's to say growth media that contains these heavy amino acids. And then you can do something to one of them, or not, if you want a really boring experiment. But let's say you treat this with a drug, you irradiate it, you do something to change the biology in one of these conditions. And then once you've done that, you can lyse the cells using one of the techniques that Monica has told you about, and mix the proteins together. Now, everything that happens to proteins from this state happens to proteins in this state. All of that sample preparation stuff happens in parallel. The reduction, the alkylation, the digestion, the cleanup. Anything that happens to one happens to both together using SILAC. It's a great reducer of technical variability in proteomics experiments. All right, so once we do that, we can do our LCMS experiment. We can find differences that are due to heavy and light amino acids in each spectra. We can make extracted ion chromatograms now of heavy and light versions of peptides that we see together, and they're coming out at exactly the same time in the LC dimension, the retention time dimension of an LCMS experiment. And so we can co-integrate areas under these peaks, which are kind of perfect matches to each other, except for the mass spacing. And we can now determine a ratio. So for this given analyte, I'm not 100% sure what it is. It looks like it had an arginine in it. And if it were charge state 1, it had an arginine in it. And then the area under the heavy curve was 80% of that under the light curve. Or you could do instantaneous point-to-point comparisons of every time you sampled along this peak and draw a line, and you can find the slope of that line, and that's basically the same thing. It's the ratio of heavy to light, and it's in good agreement with the area under the curve. So what you get at the end of the day is a ratio of a protein or a peptide in 2 or 3 conditions, depending on the plexity of your SILAC leveling, whether you just use heavy and light or heavy, medium, light. And you get the relative abundance among the conditions that you're comparing. That's how SILAC works. So what are some of the experimental paradigms that you can use with SILAC? This should look pretty familiar from some stuff you've seen before. The first is what I just described, a global protein expression experiment. Let's say you grew cells at 37, and then you heat shock cells that were grown in heavy media, and now you mix these 1-to-1, you do digest, all that great stuff that Monica told you about, and now you do your LC-MS experiment, and now for all the peptides and therefore the proteins in that sample that you're able to measure using the mass spectrometer, you can get quantitative ratios. Let's say you're interested in phosphor-proteomics. For example, you grow your cells in light and heavy. Let's do the same thing. Let's heat shock those cells, mix them 1-to-1, digest just as before. Now we're going to add in a step of enrichment, let's say for phosphopeptides, or you could do ubiquital peptides, or you could do acetylated peptides. Same thing, we can get 1-to-1, or we can get measurements of these things together. Okay. And then what about protein interaction profiling or any kind of interactomics experiment, whether it's proteins, small molecules, or nucleic acid species? Now the paradigm becomes slightly different. We still have our light and our heavy cells, and we'll try to engineer some kind of condition where the bait is absent in one situation and the bait is present in another situation. You do these IPs now or in parallel, because if you mix them at the beginning, you wouldn't be able to separate out your heavy from your light proteins anymore. But as soon as you capture all these things, you can mix them together at the sort of mid or post-IP stage, mix them together 1-to-1, digest, and now same LC-MS experiment where you measure all the ratios of all the peptides and the proteins. So those are some cool things you can do with SILAC. I bet you could probably think of more, or maybe there's some ideas occurring to you right now. We would love to hear about them. Here's one kind of really neat SILAC special case example called pulse SILAC, where now you can use 3 channels of SILAC, heavy, medium, and light, but instead of growing cells all in heavy, medium, and light, you can keep one channel constant. Let's say we'll keep these green dishes are grown in medium SILAC media, and then you start one in light media. You have it all grown in light media, and then at the time that you effect some perturbation, you also change the media. So you go, this is basically a pulse labeling type of experiment where you start out as light and then add in heavy. And then if you measure the light, medium, and heavy versions of all the proteins in the course of the time of the experiment, you can get both the synthesis rate of the new protein using the SILAC amino acid, the heavy amino acid, and the degradation rate of the old protein watching the light version of the peptide or protein disappear. And you can get a very neat sort of time course measurement that way. So that's sort of SILAC in a nutshell in one clever application of it. There are other ways that you can do metabolic labeling besides SILAC. You could do complete N15 labeling or complete C13 labeling of all proteins and peptides in cells. Usually you would do this in a sort of lower organism who can synthesize all of their amino acids starting from the very basic precursors. It's not as straightforward to quantify as SILAC because the masses that you expect between the differences of the peptides differ based on the composition of that peptide, not just how many lysines and arginines it has, but of all the amino acids. Okay. There are some times when you can't use a sort of metabolic labeling trick like SILAC. And such cases include when you have either primary cells that won't grow in cell culture or tissue, maybe a tumor sample that you're trying to characterize in some way, or a group of samples. And in this case, it's good to resort to chemical labeling. You also have chances to be able to do higher multiplexing than in SILAC at the cost of having some greater technical variability in your experiments. And you can use chemical labeling things to be able to do quantification in the same way that I just showed you with SILAC, where we take the area under the curve of heavy and light versions of it, or in another type of way where we're actually going to look at special fragment ions in fragmentation mass spectra as another option for quantification. So probably the most well-known version of the chemical labeling approach is something that's called iTRAQ, which stands for isobaric tags for relative and absolute, and I sort of put a question mark by the absolute because I'm not really sure how it's absolute, but they like to claim it is. Quantification. At the end of the day, it's quantification. So this is what the iTRAQ reagent looks like, and you can put, again, isotopic, stable isotope atoms at various positions in this molecule. And this molecule has some special properties. One is it has a group over here that reacts with primary amines, that's to say lysines and N-termini of peptides. Then it also has a group here to the left of this dotted line that likes to fall off when you do collision-induced dissociation or other fragmentation techniques. So that's good for the fragment ion quantification part. So you can construct versions of this reagent in different ways such that if you add the right number of stable isotope atoms in the right places, they all weigh pretty much the exact same thing. These four versions where I've depicted the labels, they have nominally the same mass adduct on each, right? So then, that's what isobaric means, they're the same mass. But so then we attach them on to peptides, I'll show you that in a minute. But when we fragment them, because this little part likes to fragment off, you can use this part to kind of balance out, and you can get four different fragment ions based on four different kind of formulations or positions of the stable isotopes in this iTRAQ reagent. And those happen to be M over Z 114 through 117. So very simply, how does this work? After you do a protein digest and you have clean peptides, you mix these iTRAQ reagents with your newly formed peptides that have nice N-termini, and then you can get them attached onto the N-termini. And if you had a lysine here rather than an arginine, you would get another piece of it there. And then let's say in the course of the proteomics experiment, you take a spectrum of this peptide and fragmentation spectrum of this peptide. You might see something like this, where if you look at the whole mass spectrum, you get lots of ions here that correspond to the backbone amino acid sequence of the peptide. But if you look in the region from 114 to 117 M over Z, you can see 4 ions that might correspond to the 4 different variations on the iTRAQ reagent that I showed you on the last slide. So how does this work practically now in a real biology experiment? Let's say you were testing kinase inhibitors. Let's test 3 different kinase inhibitors versus DMSO control. So now the cells, we don't need to grow them in any special media, so don't let the colors kind of confuse you here. It's just to show that they're in different, there are different conditions here, okay? So we grow our cells, we do nothing special except treat 3 of them with kinase inhibitors and 1 with DMSO. And let's say we want to look at phosphopeptides only, since we're bothering to look at kinase inhibitors, right? So we lyse and digest as before. We label each one of these conditions with a different version of the iTRAQ reagent, and I tend to call them different colors. It's kind of like 4-color DNA sequencing. We have 4 colors that we can attach to the end of the peptide, except the colors in this case happen to be masses, right? We do a phosphopeptide enrichment, we do LC-MS experiment. Along the course of the experiment, we take several MS-MS spectra, and we can see maybe some examples of what's going on. So if we took a spectrum, let's say we can identify the peptide using all of Carl's techniques, then all we need to do is look in this reporter ion region, that's sort of a buzzword, reporter ion region, to see what's going on. On a given peptide number 1, if we saw basically all the heights of these things are the same, there's no effect on this peptide when we treat it with any of these kinase inhibitors. Peptide number 2, in this case, DMSO has a high peak, and then the 3 kinase inhibitors that we tried all have low peaks. So this peptide was equally affected basically by these 3 kinase inhibitors. Or we can find things that are selective in this example here, where it seems like this kinase inhibitor 2, which happens to correspond to the 116 label channel, is not affected, whereas kinase inhibitor 1, kinase inhibitor 3 have an effect on the phosphorylation level of this peptide. We're not doing area under the curve anymore, however. We're doing, in this single spectrum, what are the heights of the ion reporters? So it's a little bit of a paradigm shift from SILAC. Yeah?
This is not protein specific, right? You're labeling the kind of dual protein that comes out of this lysine digestion.
That's right. So the question is, this is not protein specific, is it? And in fact, you don't want it to be. You want every single peptide in your protein digestion to get an iTRAQ label on it. And if you don't successfully label all of the peptides, then you have an even more complex sample, because you have 2 versions of every peptide floating around in your sample. Did you have a follow-up to that?
So would this depend on whether or not the protein is able to be labeled by these isotope tags?
Okay, so the question is, does this depend on whether the protein can be labeled by the isobaric tags? So the first thing we do is take the proteins into peptides. And when you digest the protein into peptides, you form new N-termini all the time. And N-termini of peptides are reactive with this reagent. So almost every peptide that you can imagine in a proteomics experiment is going to be reactive with an iTRAQ type reagent. An exception might be an N-terminally blocked peptide, or if a peptide has cyclized in solution before it can be labeled with the iTRAQ reagent. So, but yes, we want everybody to be labeled, for sure. Okay. All right, so there are different flavors of iTRAQ or iTRAQ-like things. There's the classic iTRAQ, which is up to 4-plex. There is something called TMT. It's made by a different company that can go up to 6-plex. There's another version of iTRAQ that is 8-plex. And in our experience, the sort of plex level is inversely proportional to the number of peptide and protein IDs. So you get more peptides out when you do iTRAQ 4-plex than TMT 6-plex, than iTRAQ 8-plex. But it's not to say that you shouldn't use these other things. There are cases where you'd really like to group 6 or 8 samples together. By all means, use these things. You'll have a little bit less depth of coverage, but you'll get all the information that you want in one experiment. Sometimes the experiment should dictate. Okay? And again, what you get out is the ratio of a peptide or an agglomeration of peptides into proteins, or a protein in some number of conditions, between 4 and 8 conditions, and it's a relative abundance. It's a ratio that you get. Okay.
Is it comparable to SILAC in terms of the protein peptide ID?
Yep. There's pretty much. That's actually quite a thorny question. So the question was, is it comparable to SILAC in terms of protein number and peptide ID? And for the most part, it is. There are some properties of the iTRAQ 4-plex reagent that actually make it slightly better than regular peptides in a proteomic experiment. They tend to retain better on a column. They tend to fragment more efficiently. Their ionization is boosted. The other thing is, if you start with 4 different samples and you label them all with an isobaric reagent, you're actually piling up the concentration of peptide at any given mass. So you kind of, if you had 4 things in equal amounts, now you've got sort of 4 times the baseline signal for that thing. That's all at the same mass, as opposed to SILAC. If you had 2 different SILAC channels, now from the starting point, you already have different masses. You're not kind of piling signal up on top of each other. You have one at the light MZ and one at the heavy MZ, and they're each maybe equimolar, but they don't contribute to each other's signal, whereas in iTRAQ, you do get that sort of thing.
So when you say relative abundance, it's relative to what?
To whatever you want in your experiment. So in this particular experiment, the way that we've designed it is to say, all right, we have these 3 conditions that we care about, and this one that's our control condition, and that's relative to that. So we would take literally the ratio of intensity at, say, 115 to 114 and say that is the relative enrichment or decrease of the given phosphopeptide for kinase inhibitor 1 over control. It's however you design the experiment, but always an experiment should have a good control. Okay, so there are other ways that you can label things chemically. There's a technique called reductive dimethylation. We talked a lot before about not wanting to carbamylate things unless you do, because you can get stable isotope versions of urea and intentionally carbamylate everything and use that as a sort of poor man's stable isotope label. You can do O18 labeling, in fact using trypsin to introduce a heavy O18 atom on the C-terminus of peptides, and or any kind of amine or sulfhydryl reactive reagent that you want where you could make stable isotope versions of it to do a kind of chemical version of SILAC quantitation. Okay. I'm just going to touch very briefly on this because we're going to hear a lot more about it later. You can always, if there's something that you care very deeply about, both observing and getting very good quantification on, you can make a peptide in the laboratory or a contracted company to do it that already has a stable isotope amino acid incorporated into it. You can use this for both precursor and fragment-based quantification, and Hosmek will talk more about that next. When you spike a peptide into the sample, you can be pretty much guaranteed that you're going to observe it. In fact, you can target it using the mass spectrometer. So if it's something that you need to see every time in a series of experiments, you might consider doing this. And then you can spike a known amount. Since it's synthetic and it comes in a test tube, you can just weigh out a certain amount and spike the known amount into your sample, and that gets you more towards absolute abundances of the analyte in your sample. Okay. I'll touch very briefly on spectral counting. So the mass spectrometer is a sampling instrument. It has to sort of pick peptides that it's going to sequence on the fly. It picks a precursor, fragments it, and reads it out. But it's not comprehensive. We don't sequence all of the peptide peaks in an LC-MS experiment. We leave some behind. We know that because if we redo the same sample multiple times, we find new proteins. So we know that we're not comprehensive, but still, your likelihood of sampling a given peptide or a peptide from a given protein is tied or somehow proportional to its abundance. There are a lot of caveats to this. Different instrument vendors have different ways of picking which peptides are going to be sequenced, how many peptides there could even be from a given protein. There's all kinds of things that could hang you up on here. But the basic premise of spectral counting is that one spectrum with a peptide spectrum ID match that can be linked to a protein equals one count for that protein. And that's essentially spectral counting. We tend not to practice this here. It has appeared in sort of protein expression profiling and biomarker discovery efforts from other labs. But again, it's hard to kind of generate statistical models to figure out if this quantification is real. It's hard for us to do an experiment in our laboratory and ask another laboratory to reproduce the same result that we got. You don't get a quantitative ratio. It's really, it's a mess, and I wouldn't recommend it for serious quantitative proteomics experiments. Okay. This is just in your handout. It goes through all the kind of pluses and minuses of these various techniques along with some application examples. And so you can go through that on your own. So now we have ways to quantify peptides. And how do we roll this up into quantification of a protein? So let's take the simple case where we observed a whole bunch of peptides from a single protein. And we have observed SILAC ratios for all of these peptides, again, belonging to that same protein. We can just use simple descriptive statistics. What we tend to do is to pick the median value peptide from a list of peptides that belong to a single protein. So in this case, we would say something like the median of these peptides, and I've taken the log to the SILAC ratio, is basically close to 1-to-1. 0 would be 1-to-1. Okay. Then there are cases like this where, and Carl had illustrated this before, where we have a list of peptides that belong to one protein, some other peptides that belong to another protein, Y, and then one peptide that can go either way. It belongs to both of these proteins, or at least in theory could. What we tend to do is to put this peptide with the protein that has more evidence. This is a sort of Occam's razor approach to explaining the existence of this peptide. So if we were going to try to figure out the ratio of this protein X, we would count this one as a member of protein X, but we wouldn't count it as a member of protein Y. And because we applied this Occam's razor principle, meaning the bulk of the evidence supports that it belongs to this protein, we would call this a razor peptide. Okay. And then there are cases where you have isoforms. This is like the human and mouse experiment analog that Carl presented. There's protein X has isoform 1 and 2, and there are some peptides that are unique to isoform 1 and some that are unique to isoform 2, but a whole bunch that are in common. If you really want to quantify what's in or what are changes in isoform 1 and isoform 2, you have to throw out all those that are in common and then pick only the ones that are unique and specific to the isoforms that you're interested in. So you get kind of poor statistics because you throw out a lot of evidence in this case, but if that's what you're after, that's what you have to do. Okay? And then ultimately, at the end of the day, what do you get out from a quantitative proteomics experiment? Should be a table of values that's sort of organized by proteins. Each row is a protein, and it should have some more descriptive information about it, maybe a gene name for it, or a list of accessions, a uniprot accessions where you could find out more about that protein. And you should get a ratio and perhaps also a range around that ratio. I haven't illustrated it here, but that's something that we could report. And then if you've designed your experiment in such a way that's comparing 2 or more conditions, and you're interested in knowing what changes from condition 1 to condition 2, we could give you, using some descriptive statistics models that Mani will talk about tomorrow, a sort of false discovery rate. Now, not that we've identified that protein, but that protein is changed from condition 1 to condition 2. So you've got 2 sets of false discoveries that you might be able to arrive at the end of the day. So what it really boils down to is we're going to hand off to you a prioritized list, whether it's by the SILAC ratio or iTRAQ ratio or false discovery rate, prioritized list of candidates that should inform your next experiment. Proteomics is usually not the final experiment in a big study. It's usually near the beginning, and we hope to provide you with direction through the type of experiments that we do. So at the end of your packet, there are some references to find out the sort of primary literature on some of these techniques. But that's it from me, and if there are any questions, I'd be happy to take them. Yeah?
So, the peptide to protein quantification, you were doing that for a phosphoproteomics, and presumably, like some phosphosites are differential and others are not. Do you use that information to infer potentially the abundance? Those information be used for protein level analysis?
Okay, so the question is, how do you kind of roll up peptides to proteins and use it for quantification in a phosphoproteome experiment? So that's a little tricky because remember, in a phosphoproteome experiment, a lot of what we do involves something that has a phosphopeptide enrichment step. So in a straight phosphoproteomics experiment, the bulk of the peptides that we measure are phosphopeptides. And in that case, the quantification that we report is at the site level, especially given the caveat of how well we've localized that site. Now we can do a more comprehensive experiment where we can measure alongside of the phosphoproteome the global proteome. And if we are lucky enough to be able to observe a phosphopeptide and the non-phosphorylated version of that peptide and a third peptide from that same protein across multiple conditions, then we can actually infer the stoichiometry of that site. So. But mostly the goal in phosphoproteomics experiments is to report the differential abundance of given phosphosites. Okay, well, thanks.